Hello and welcome. Today let's talk about Chapter 25. Chapter 25 is going to deal with DNA structure and gene expression. So first we'll go through the structure of DNA, look at the whole structure of DNA, and then we'll move on to gene expression. So when we talk DNA structure, I'd like you to know in the mid-20th century, geneticists were determining that DNA, which stands for deoxyribonucleic acid, is the genetic material. And biochemists were determining the structure of DNA. These research efforts led to our knowledge of modern molecular biology. The structure of DNA was determined by James Watson and Francis Crick. They determined the structure of DNA in the early 1950s. DNA is a chain of nucleotides. Each nucleotide is a complex of 3 subunits. We'll see we have a phosphate, phosphoric acid. We have a deoxyribose, our pentose sugar. And we have a nitrogen-containing base. So what I'm going to do is I'm just going to jump a little ahead and I'll show you exactly that. So here we have a nucleotide. We said each nucleotide is a complex of 3 subunits. You've got your phosphate, you've got your deoxyribose sugar, and then you've got your nitrogen-containing base. Same thing here with this one. Here you've got your phosphate, your sugar, and then your base. There are 4 possible bases. There are 4 possible bases that we're going to find in relation to DNA, and they will include our 2 purines, which are double ring structures, and they include adenine and guanine. Adenine is abbreviated with a capital A, guanine is abbreviated with a capital G, and our 2 pyrimidines, thymine, abbreviated with a capital T, and cytosine, which is abbreviated with a capital C. Thymine and cytosine are going to be single ring structures. They have only 1 ring to their structure. Our purines, they have 2 rings to their structure. One way I remembered my purines was they are pure as gold. Pure as gold. So one way to remember, and the other. Adenine and guanine, again, are our purines. Thymine and cytosine are our pyrimidines. A DNA polynucleotide strand has a backbone that's made up of alternating phosphate and sugar molecules. So here we can see that. Right in here, it's back in here. This is the sugar phosphate alternating backbone. And then that's with that one, and the same thing here we have with this one. So let's look at it in greater detail. Here you can see phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So you have your backbone made up of alternating phosphate and sugar molecules on both sides. The bases then we see here, the bases are attached to the sugar, but they project onto one side. The bases are attached to the sugar, but they project onto one side. Attached to the sugar, projecting onto one side. Attached to the sugar, but projecting onto one side. Attached to the sugar, projecting onto one side. DNA has 2 strands that twist about one another to form a double helix. So here you can see that very well. DNA has 2 strands that twist about one another to form a double helix. The strands are held together by hydrogen bonds. They are held together by hydrogen bonds between the bases. So right inside of here where we're going to find the hydrogen bonds. So let's move back here. Here you can see between the bases the hydrogen bonds. Adenine always pairs with thymine. And they're held there by 2 hydrogen bonds you can see. Guanine always pairs with cytosine. And they're held together by 3 hydrogen bonds. Notice that a purine is always bonded to a pyrimidine. So we call all of this complementary base pairing, where adenine is always going to pair with thymine and guanine is always going to pair with cytosine in DNA. So you see purine and pyrimidine, purine and pyrimidine. You don't have adenine binding with cytosine or thymine binding with guanine. We have complementary base pairing. And then our two strands are said to be anti-parallel orientated in opposite directions. And we'll see why that is. We're going to see here the sugars are orientated differently. There is a 5' free end on one sugar and a 3' free end on the other sugar. So here we can see that. So here we can see we have our one nucleotide. We've got our phosphate, our sugar, and here we've got our base. Now what we're going to do is we're going to number the carbons on our sugar. So when we look at the carbons in greater detail, so here we can see we will number our carbons. So here we've got our 1' carbon, our 2' carbon, our 3' carbon, our 4' carbon, and our 5' carbon. So attached to the 5' carbon is always the phosphate. Attached to the 1' carbon is always the base. And then the 3' carbon we're going to see is going to bind with then the next phosphate. Here it's empty. Here it's empty. So here on this side you've got the 5' end of this sugar, so we refer to this as the 5' end of this molecule. Here we have the 3' end of the sugar, so we refer to this as the 3' end of the molecule. And here you can see these two strands are found in an anti-parallel orientation. You have a 5' end here and a 3' end on this opposite side. And then here we have 5' end on this side and a 3' end on this side. If I was to give you a nucleotide sequence and ask you to give me the corresponding sequence, you should be able to construct that then with this knowledge. So if we were to move back here to one slide, just because there's more space here and I can write here, if you were to see, if we had this sequence, let's say for example, A, T, G, C, A, C, T, and ask you to give me the matching sequence to this, then you would have to give me, right, we would have T, A, C, G, very good, T, G, A. Okay, so by having this basic knowledge of complementary base pairing. And then you can see that here, A and T, G and C, A and T, G and C, A and T. So if we were to hide this strand on this side here, let's say if we were to hide this strand here, cover it up, let you see only this side here, and ask you then, give me the complementary strand to this strand here, so then you would be able to do exactly what we see here, right? So if this A was covered up, you would be able to figure that out. Across from the T would be A, across from the G, C, A, G, and then T. And that's exactly what we see here. All right, let's move then to DNA replication. Now, when we talk DNA replication, here we're going to see basically one strand being copied. And it's going to be copied so that way we can take those instructions that are encoded in that certain strand and encode certain proteins for that segment. So here, when we talk DNA replication, when the body grows or heals itself, cells are going to divide. Each new cell that's going to be formed is going to require an exact copy of DNA that the original cell contained. DNA replication is the process of copying one DNA double helix into two identical double helices. This process is going to be carried out by an enzyme called DNA polymerase. So DNA replication is the process of copying one DNA double helix into two identical double helices. This process is carried out by DNA polymerase. DNA polymerase is the key enzyme in DNA replication. It's going to use the original strand. DNA polymerase uses the original strand as a template for the formation of new strands. DNA polymerase is that enzyme. So you can see that right inside of here, DNA polymerase. Now, semi-conservative replication uses each original as a template to produce new complementary strands. Semi-conservative replication is going to use each original strand as a template to produce a new complementary strand. Now, when we talk semi-conservative replication, here in semi-conservative replication, each daughter DNA molecule consists of one new chain of nucleotides and one from the parent DNA molecule. So you got to remember that. So each daughter DNA molecule is going to consist of one new chain of nucleotides. It'll consist of one new chain of nucleotides and one from the parent DNA molecule. The two daughter DNA molecules will be identical to the parent molecule. They will be identical to the parent molecule. Now here, let's look at this process of DNA replication in greater detail. Now, before replication begins, the two strands of the parent molecules are hydrogen bonded together, if you recall. They're going to be hydrogen bonded together. Next, then we'll see the enzyme DNA helicase. DNA helicase is going to come into the picture. It's going to unwind and unzip the double-stranded DNA. It unwinds and it unzips the double-stranded DNA by breaking the hydrogen bonds that we saw in between the bases. Then next we can see here, new DNA nucleotides are going to be positioned and joined by the enzyme DNA polymerase. By the enzyme DNA polymerase. So DNA synthesis then occurs. Right? New DNA nucleotides fit into place along separated strands by complementary base pairing. Now, I want you to know the leading strand is going to follow the enzyme helicase. Here, let's look at that. So here's a nice picture. It depicts to us the two strands that have been opened up, and they get opened up, and you can see one strand is making its way this way, and it's being copied, and the other strand is making its way this way, and it's being copied, and then eventually at the end, we're going to have DNA replication occur where you've got two DNA produced now, versus where we had just one. So the DNA here replicates to produce two exact copies of itself. So here, let's look at this then in greater detail now. So again, here's the two strands. They've been opened up. The enzyme helicase this enzyme helicase is busy opening them up. And here you can see is one strand. That's one template strand. And then here we have the other template strand. So here we can see our two strands. And these two strands are being opened up by this enzyme helicase. And here we can see our template strands split open. Here's one template strand, the other template strand making its way this way. And now here on the leading strand, DNA replication is, we said, continuous. Here we're going to have this leading strand, and this leading strand is going to follow this enzyme helicase. While synthesis on the lagging strand is going to result in the formation of short segments of DNA that are called Okazaki fragments, you see here. Then this enzyme, DNA ligase, comes into the picture. DNA ligase is going to help connect the Okazaki fragments, and it seals any breaks. And you can see there, then we're going to have produced two double helix molecules, which are going to be identical to each other. Chemotherapeutic drugs for cancer treatment stop DNA replication and cell division. Let's move to then gene expression. Let's actually see then the genes being expressed that are here on these DNA segments. So here then let's move to gene expression. Now, when we talk about gene expression, here in gene expression, this is the process of using a gene sequence from our DNA to synthesize a protein. This process is going to depend on different types of RNA molecules. RNA now stands for ribonucleic acid versus DNA, which was for deoxyribonucleic acid. Now, when we talk RNA, we have three different types of RNA molecules. First, we have mRNA, messenger RNA, tRNA, transfer RNA, and third, ribosomal rRNA. So next then, let's compare DNA and RNA, and then we'll jump into the actual process of gene expression. So here we can look at this table. This table compares DNA and RNA. Now when we talk about the sugar each of them contain, DNA is going to contain deoxyribose as its sugar, while RNA is going to contain ribose as its sugar. Now the bases we're going to find in relation to DNA are, as we mentioned before, adenine, guanine, thymine, and cytosine. Now when we talk RNA, we're going to see in place of thymine, we have what we call uracil. So we'll have adenine, guanine, uracil, and cytosine. RNA, you can see, is a single-stranded structure, while DNA is double-stranded. And here we can see DNA is found as a helix, while RNA is going to be found as, again, single-stranded structure, while RNA will not be found as a helix. When we talk about gene expression depends on two processes. The two processes are termed transcription and translation. Now, when we talk transcription, I would like you to know takes place in the nucleus. During transcription, a segment of the DNA called a gene serves as a template for the production of an RNA molecule. So here we'll see an RNA molecule being produced. A gene is a genomic sequence, segment you can think of on DNA, that directly encodes for functional products, either RNA or for proteins. We will focus on transcription to form messenger RNA, mRNA, which is the first step in protein synthesis. So let's talk then messenger RNA. Now, when we talk about messenger RNA, I'd like you to know the purpose of messenger RNA is to carry genetic information from the DNA to the ribosome for protein synthesis. Messenger RNA is formed by the process of transcription, which is going to take place inside of the nucleus, Transcription begins when an enzyme called RNA polymerase binds tightly to a promoter region. A promoter region is going to be a region of DNA that contains a special sequence. This enzyme opens up the DNA helix so base pairing can occur. It inserts RNA nucleotides, right, because we're making an mRNA. It inserts RNA nucleotides, and an mRNA molecule is going to result. When mRNA forms, it has a sequence of bases complementary to that of the DNA. Wherever A, T, G, or C are present in the DNA template, U, A, C, G are going to be found on the mRNA molecules. mRNA is a faithful copy of the sequence of bases in our DNA, but we have a uracil in place of thymine. So we can see here, our messenger RNA serves to carry genetic information from our DNA to our ribosomes for protein synthesis. I'd like to think of the cell, when you think of the cell, I like to think of the cell as a factory. What do factories do? They produce things. Here, one function we're looking at of our cells is protein synthesis. Now, DNA is, I like to think of as the boss in this factory. DNA is the boss, and DNA gives the directions of how the proteins need to be made. DNA sits inside of its office, which is the nucleus. mRNA I like to think of as the messenger, right? The manager you can think of. The manager gets the directions from the boss and brings those directions to the ribosome, to the floor in the warehouse, in the factory, for production, for protein synthesis then. So the messenger RNA you can think of as a manager. Goes to the DNA, gets the instructions for what needs to be done, and comes out to then the ribosome where it'll have everything basically taken care of. So mRNA is formed by the process that we call transcription. So that's the whole function of transcription, to produce an mRNA molecule. Now this process is going to begin when RNA polymerase is going to bind to, we said, a promoter region on the DNA. The DNA helix then is opened up so complementary base pairing can occur. The RNA polymerase is going to join the new RNA nucleotides in a sequence that's going to be complementary to the DNA, but we said the difference is going to be in place of thymine, we have uracil. So let's look at this. So here you can see now we've got our DNA double strand. Now we want to open up just this area here. So this is where the information is at that we want to copy. So here we can see the strands are opened up. And now here, this is the segment that we want to copy. So here then our RNA polymerase is positioned, and RNA polymerase starts to copy everything complementary to that segment there. So we had thymine here, adenine is placed there. Guanine, cytosine. Cytosine, guanine. Adenine, we don't have thymine, uracil we said. Guanine, cytosine. So here you can see we're making a strand with basically the instructions from our DNA strand. So this is what our mRNA is, right inside of here. Now this mRNA, once it gets constructed, right, once all of the information is put on the mRNA is going to get processed. It undergoes processing. So when we talk about processing, okay, so here we saw with our messenger RNA, everything here, and then everything right inside of here. Okay, that's what brought us to this picture. So here then we have processing that's going to occur. Now when we talk processing, we've made the mRNA. However, the mRNA has information on there that we don't need, and it needs to be basically fully marked. So what we're going to do is we're going to process it. Now here, what happens is after the mRNA is transcribed, right? This process is called transcription. Transcription, if you think transcription, what do transcriptionists do? Transcriptionists will take a message that they'll hear and they'll type it out. Physicians use transcriptionists all the time when they're seeing patients. They don't have time to sit down in between patients and write notes. So what they'll do is they'll record it on an audio recorder, and they'll give that to a transcriptionist, and the transcription will listen to the audio recording and type out everything that has been said. So here, basically, that's what we have done. We have typed out everything basically that DNA is saying in the form of an mRNA. But that mRNA has a lot of information that doesn't need to be there, so we're going to process this mRNA. Now here, when we talk processing, so it must be processed. This mRNA must be processed before entering the cytoplasm and making its way over to the ribosome. Now here we'll see this newly synthesized primary mRNA molecule must undergo processing to become a mature mRNA molecule. Most genes in humans are interrupted by segments of DNA that are not a part of the gene. These portions are called introns. The other portions of the gene are called exons, and they are excised away from the introns because they're going to be expressed. Only exons result in protein products. So here we can see our primary mRNA is going to contain bases complementary to both introns and exon segments of DNA. But the introns are intragene segments which get removed because they're not going to code for proteins. Exons are going to be the portions of a gene that is actually expressed. Also during processing, Also during processing, we'll see one end is going to receive a guanine cap. The other end is going to receive a poly-A tail. So the 3' end is going to receive a poly-A tail. That's a lot of adenines. Just adenine, 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 adenine. So they refer to that as a poly-A tail. Then the mature mRNA molecule is going to be ready to make its way over to the ribosome for the next process called translation. So here we can see then our DNA molecule, and this is the region that we want to transcribe from here to right inside of here. So let's say we transcribe this region. You can see this region has both introns and exons. So if we transcribe that region, we make this mRNA molecule that we've got. This mRNA molecule then has to undergo processing, we said, where we're going to add a cap and we're going to add a poly-A tail. And here you can see we've got our introns in here. We want to remove those introns because they're going to be non-coding segments. One way I remember this was exons are excised away from the introns and they are expressed. So you have everything EX, EX, EX. So here you can see we've got introns and exons. We've got the cap added in our poly-A tail. Next then here you can see we'll cut out the introns and we're going to connect all the exons in the same order as we found them before. They'll be connected in the same order as they were found before. And here you can see then we have our mature mRNA molecule. Now this mature mRNA molecule is going to be ready for translation. Next, when we talk translation, think translation. Translation, we're going to go from one language to another, basically. Right? We're going to translate this language of nucleotide sequences to this segment of amino acid sequences. So when we talk translation, translation is a second process by which gene expression is going to lead to protein synthesis. So it's a second process by which gene expression leads to protein synthesis. Translation requires several enzymes and different types of RNA molecules. So we've mentioned all 3 types of RNA molecules. So here then first I'd like to talk to you about the genetic code. When we talk about the genetic code, I want you to know the sequence of bases in DNA is transcribed into mRNA, which codes for a particular amino acid sequence to form a polypeptide. I want you to understand a triplet of nucleotides is called a codon. A triplet of nucleotides is called a codon. The triplet consists of any of the 4 bases found as 3. So triplet code is basically where each 3 nucleotide unit of an mRNA molecule is going to be called a codon of an mRNA molecule. Understand a triplet of nucleotides is called a codon. A triplet of nucleotides is called a codon. The triplet consists of any of the 4 bases found as 3. This sequence of 3 nucleotides will code for a certain protein. Since there are 4 nucleotides and 3 places, this equals to 64 possible codes which will code for certain amino acids. Now of the 64 codons, 61 are going to code for amino acids, and the remaining 3 are going to code for stop codons. And the remaining 3 will code for stop codons. Those will be U, G, A, U, A, and U, A, G, which are going to signal polypeptide termination. So they are going to code for stop codons. Here, let's look at that. So here we can see our 64 possible codons. For the first base, we'll use this section here. Second base will come from this section here, and then third base will come from this section here. So let's say, for example, we have AUG. Here's A, U. So we're going to come down into this column here. So we have A, here we have U, and then we're going to look for G. G comes right down in here. So here's AUG, this possible codon. AUG, you can see it codes for a start codon, which is methionine. And here you can see then we've got UAA, we said, UAG, and UGA, which all code for stop codons. The way I remembered them was you are away, you go away, or you are gone. One way to remember the stop codons. Okay? You don't have to actually remember each and every codon and which amino acid it's going to code for. No. So next then let's move and let's talk about transfer RNA, tRNA. So let's talk tRNA, transfer RNA. tRNA is going to be important because tRNA is going to transport amino acids to the ribosome where protein synthesis is taking place. Each tRNA molecule has on one end an amino acid. It has on one end an amino acid, and the other end is an anticodon end, a triplet of 3 bases complementary to a codon of mRNA. Now, when the tRNA amino acid complex comes to the ribosome, its anticodon is going to pair with an mRNA codon. So when we talk tRNA, tRNA has a boot-like shape to its structure. So one end we said is going to be an anticodon end, and the other end is going to be the end that contains a specific amino acid. So we said one end will have an amino acid, and the other end will be the anticodon end, where it's going to have a triplet of 3 bases complementary to a specific codon of mRNA, we said. So here we can actually see that tRNA. So here you've got your tRNA, here's the anticodon end, and here's the amino acid end. The anticodon end you can see here in greater detail then. It's going to be complementary to the mRNA sequence. So here then is the amino acid it'll carry. So here we could see now basically these processes summed up in a picture. We've got our DNA helix, our double helix. The helix gets broken apart basically. We copy one segment of it. So here we can see is that segment that we copy. And we make an mRNA complementary to that strand. This mRNA then has codons, you can see. These codons then are going to code for specific amino acids. And here we can see then in this process of translation, we'll attach those specific amino acids to those certain codons, and voila, we'll have our polypeptide being formed. So next then let's talk about ribosomes and ribosomal RNA. When we talk ribosomes and ribosomal RNA, they're going to be formed by the nucleolus. They're formed by the nucleolus. Ribosomes are going to be small structural bodies. Ribosomes are small structural bodies found in the cytoplasm and on the endoplasmic reticulum where translation is going to occur. A ribosome has a binding site for mRNA as well as binding sites for 3 tRNA molecules. I like to think of this process now as a party, right? The party is going to happen and we invite guests to the party. mRNA is a guest, tRNA is a guest, and the party is happening at the ribosome's house or the rRNA's house. So mRNA is invited, tRNA is there, all the RNAs are there. So when ribosomal RNA is having a party, ribosomal RNA has to accommodate the guests and have seats for them. So you can think of that basically being the binding sites for mRNA and then binding sites for tRNA as the seats for them. As the ribosome moves down, these binding sites, they facilitate complementary base pairing between the tRNA anticodons and the mRNA codons. Binding sites are also going to bring amino acids in line in specific order to help form the polypeptide. So we'll see after the initial portion of mRNA is translated by one ribosome, the ribosome begins to move down the mRNA. Then other ribosomes may attach to the mRNA. Several ribosomes may move along the same mRNA. Multiple copies of a polypeptide may be made sometimes. The entire complex is going to be called a polyribosome then. A polyribosome. So here we can see the structure of our ribosomes. You have a large subunit and you have a small subunit. The two subunits come together to form your ribosomes. So here then what we'll do is we'll have the sites added to this. So here you've got your tRNA binding sites, and here we can see basically the mRNA binding site underneath here. So it accommodates mRNA, and the tRNA binding sites will accommodate tRNAs, 3 of them. So we'll see here the sites are called the E site, the P site, and the A site. The E site is for the exit site. P site we'll see is the peptidyl site. And then the A site is the aminoacyl site. Now what's going to happen here is our incoming tRNA is going to come and sit in this A site. And then the tRNA that's sitting here in the P site, it's going to give the growing protein up to this new tRNA. And now it's going to be empty. So then that tRNA is going to jump into this E site, where then it's going to eventually exit. Then the tRNA that's sitting in the A site, now it has the growing polypeptide chain, it's going to jump into the P site. So you can see the P site is going to contain the growing polypeptide, the A site is for the incoming tRNA, and the E site is for the outgoing tRNA. It's going to be kind of like a organized musical chairs here. So we'll see here then, as this ribosome moves down this mRNA, new tRNAs are going to arrive and a polypeptide is going to form and grow longer. So here we can see that. So here we've got our ribosome, and here it's on this segment of the mRNA. And here you can see is our growing polypeptide chain. So here is the tRNA. The tRNA is seated in the P site, and it has that growing polypeptide chain I told you. Now the A site is empty, and the E site is empty. Now what happens is a new tRNA comes into the picture and it sits into, we said, the A site. And here you can see it's got its amino acid there. When it sits in the A site then, what happens is the tRNA that's in the P site, I told you, gives up its growing polypeptide chain to the new tRNA. It says here, you hold on to it. It gives it to the new tRNA. The tRNA adds it to its own amino acid. Now this tRNA that's in the P site, now this tRNA in the P site is going to be empty, you can see here. The tRNA in the A site has the growing polypeptide chain. Then this tRNA in the P site is going to jump into the E site. Once it jumps into the E site, then it's going to be able to exit from there. And that tRNA in the A site is going to jump into the P site. So here you can see it's in the P site. So here now we are back at where we started. Now this is going to happen all over again. A new tRNA is going to come in. So the new tRNA comes in. This P site tRNA is going to give up its growing polypeptide chain. So it gives it up to then this new tRNA. And then it's going to be basically empty. And it's going to jump into the E site. And then it's going to take off. And then this A site tRNA is going to jump into the P site. Ready to begin the process again. So that's why I described it as like a organized musical chairs. So we're taking this language of nucleotides we see on this mRNA and converting it to this language of amino acids. And converting it to this language of amino acids. And eventually proteins. Translation terminates once the polypeptide is fully formed and a stop codon is reached. So it's going to keep occurring until we reach UGA, UAA, or UAG. And once we reach those segments, everything will break apart, and that protein then is going to take off into the endoplasmic reticulum. So when we talk translation, translation is going to require 3 steps. First, you have initiation, then you have elongation, and third, we're going to have termination. So during translation, codons are going to base pair with anticodons carrying specific amino acids. The order of codons determines the order of tRNA entering the ribosome and the sequence of amino acids in a polypeptide. And the process must be orderly. So we talk translation, it involves 3 steps: initiation, elongation, and termination. We can see the first 2 steps are going to require energy. They will require energy. So let's look at initiation first. So let's look at initiation. Initiation is going to be the process that's going to be bringing all the translation components together. Initiation factors are going to assemble ribosome components. The small ribosomal subunit, the mRNA, the initiator tRNA, and the large ribosomal subunit all are going to get put together. The initiator tRNA attaches to the start codon, AUG, that segment we talked about. The ribosome has 3 binding sites, we said, for tRNA. P site stands for peptide site, A site, the amino acid site, and E site, the exit site. Then we talk elongation. Elongation, we saw, is the process by which the polypeptide increases in length. By which the polypeptide is going to increase in length. This occurs 1 amino acid at a time, right? We saw tRNA bringing in 1 amino acid at a time. Bringing in 1 amino acid at a time. In addition to tRNA, proteins called elongation factors are also required. And what they do is they facilitate the binding of tRNA anticodons to mRNA codons at the ribosome. Elongation, we'll see, is going to consist of 4 steps. First, we'll see a tRNA with attached peptide is at the P site, and a tRNA carrying the next amino acid is arriving at the A site, we said. Once the tRNA is in place at the A site, the peptide chain will be transferred to this tRNA we saw. Energy, we'll see, and part of the ribosomal subunit are going to be needed to bring about this transfer. The energy contributes to peptide bond formation, which makes the peptide 1 amino acid longer by adding the peptide from the A site. And number 4, we said translocation is going to occur. The mRNA moves forward 1 codon length, and the peptide-bearing tRNA is now at the ribosome P site. Next, then, translocation is going to occur. The mRNA moves forward 1 codon length, and the peptide-bearing tRNA is now at the ribosome's P site. The spent tRNA is going to exit then. The new codon then is going to move to the P site. It's going to move to the P site. And then the A site is going to be empty and ready to receive the next complementary tRNA. This will take us then to termination. When we talk termination, this is the final step in protein synthesis. The new polypeptide and the components that carried out protein synthesis are separated from one another, we said. We said they're going to break apart. They're going to be separated from one another. Termination of polypeptide synthesis occurs at a stop codon. Termination requires release factor proteins, which cleaves the polypeptide from the last tRNA. The ribosome then dissociates into 2 subunits. So here you can see initiation. We've got AUG found by our initiator tRNA. And here you can see the mRNA is in place then. Our tRNA is in place, all on the small ribosomal subunit. Then the large unit comes in and locks everything in place. And here, as it locks everything in place, you can see now that initiator tRNA is in the P site. Our A site is empty, ready for the incoming tRNA. So then elongation happens, right? We're going to elongate the chain, we said. So when we talk elongation, now you can see our chain is elongating. Now the new tRNA comes in, it occupies this A site, and here this growing polypeptide chain is going to be given up. It gets given up, and here you can see then, once it gives up that growing polypeptide chain to this new tRNA in the A site, that tRNA is empty. It's going to jump into the E site where it'll eventually exit, and this tRNA in this A site is going to be able to move into the P site. So you can see that happen. The polypeptide chain is given up. That tRNA is going to exit, and then our tRNA with the growing polypeptide chain that was in the A site is able to move into that P site because it's empty. And then a new tRNA will come in. So you can see here we are elongating our protein chain. Finally then, we reach a stop codon, UGA, and release factor gets produced, and here then termination is going to occur. We said everything is going to separate, and then here our protein is going to make its way into the endoplasmic reticulum. So here we have a review of gene expression. Transcription first, which takes place inside the nucleus, and then translation second, which takes place then in the cytoplasm. So I told you, think of this as a factory. It's got a job to produce things. Those instructions come from DNA, which is the boss that's housed inside of the nucleus. You can think of the office. And then our messenger RNA, our manager, is what brings the instructions for the type of protein that we need made from the DNA out to the ribosome where the protein will actually then be made.